We have a gentleman who you also think highly of up next, Dr. Robert Faust. He's been a wonderful guest for us before on the podcast, and we're happy to have him here as part of this year's conference.
I want to say one thing, Dr. Faust. In my career, such as it was, going back over 12 years, I guess, online, I only promoted one, maybe 3 companies, and he was the first and remained the last. I got to know his work, his products. I've been his ear many times on the phone, and Ryan Zato, his assistant back several years ago. Nothing but good things to say about Dr. Faust and his humic and fulvic acids and the knowledge that he spread. And I still take his Wu Jinsan, the liquid form of fulvic acid for human food grade. So he's the real deal.
Oh, awesome. Well, thank you so much again, Coot, for taking the time to join us. And thank you, Dr. Faust, for joining us again for the conference.
You bet. Can you hear me now?
Yep, now I can hear you.
Okay, good. Well, that was some kind words from your last speaker, who I know pretty well over the years. And he knows what he's talking about. That sounds self-serving, but no, in general. So what I wanted to talk about today is some of the research that we've been doing out here in Oregon, in the Willamette Valley. Because I try to get down to the nitty-gritty on things and answer questions, and and not try to base it on sort of internet rumor or let's say tradition, although I also believe traditional practices. As an agroecologist, I go back to the traditional practices. Okay, how do they farm, for instance, in Hawaii, when there was no contact at all with any other part of the world? There was no imports, there was no fertilizers, there was no pesticides, and they did it for thousands of years. Also, the inhabitants of the Willamette Valley here in Oregon, how do they do that? Okay, so that was always an interest with me. You know, if you compare today to industrial style agriculture, where it's just spray and pray on a schedule, you know. So my whole for the last 50 years, I'm trying to find the alternatives to toxic technology. So how can we get around poisoning ourselves and the earth? And that's the question. So there's certain things that we're going to do. I'm going to try to I'm going to go into one of my research papers, and it's on the subject of how can we manipulate cannabinoid distribution in hemp and cannabis plants. In other words, what can we do to influence that, either in one way or the other? Can we reduce THC levels? Let's say if we're a hemp producer and we're controlled, you know, by regulations, sort of mindless regulations on percent THC. And so we have to keep THC down. Now, I did this work after a friend of mine got his whole crop destroyed because he was like 0.35, 0.4 or something like that. So how can we control that? Then on the other hand, how can we make THC higher? Yeah, the opposite direction. Okay, so this is a research project we did here. Open field. I mean, Oregon's a great place for this. This is large scale research, and I'll show you some pictures here. So let me see. Oh, how do we do that? Okay, I'm gonna have to share the screen. Oh boy. Okay. I'm not sure.
You just have to hit a share screen, and then you choose the window that you want on the Zoom.
Yeah. Oh boy, it's not working out. I'm seeing, I can't seem to get that to do what I want.
What is it that you're trying to open? Is it a PowerPoint or photos or?
Yeah, it's a PowerPoint. It's a what do they call Keynote? Should work. Done it before.
Yeah, you should just be able to hit share screen and then and choose the browser or just the full screen. Sometimes it's easier to do that.
Yeah. So full screen. I thought I had this figured out.
Where's?
Yeah, I'm getting a screen all right. I'm not getting what I want. Oh, Keynote. There you go. So the whole idea here is to.
You could email it to me. I can share it if that's easier too, if it's not working for you.
Well, it should be working for me. Okay. Yeah. Yeah, it's a Keynote. Damn, I thought I could do the screen sharing here.
It should just be able to hit share screen or Alt S on the keyboard. Should get you to that right menu.
All what? Oh.
Hold Alt and S, and that should open the menu. Oh, if you're on a Mac, Fumidor might be able to help you better, or Marty. I don't know if Fumi's there. He's more familiar with Mac than I am.
To share.
It should be working, honestly. I don't know if you can hear me right now, but it shouldn't be any different. Press the share screen button. The sharing's enabled. There shouldn't be anything super different about it. I don't know. I can't be helpful right now.
Okay.
Yeah.
I wasn't sure.
You could again, you could email one of us, and then basically we could share it for you.
Okay. All right. I'll just keep going then. I'm not sure what.
Perhaps you have to toggle between when you click the Zoom on the Zoom window. When you click the share button, a little window comes up and it'll show like desktop, whiteboard.
Yeah,
blah, blah, blah.
yeah.
You might have to
Right.
toggle between those to get, let's say, to the basic and toggle to the right tab, and make sure that your Keynote is open. So if you're looking for the Keynote, make sure that that's open on your desktop somewhere.
Yeah, it is.
I don't know if that helps.
Okay, so Keynote's open. All I'm getting is open system preferences.
You should, when you hit share screen, it should pop up and then it should show you screen 1, screen 2, whiteboard, iPhone, iPad, and then whatever browsers that you have open or Windows.
Okay.
And you can always,
work.
yeah, you can always hit desktop 1 and then just.
All right, now, okay, it's going to work now. Okay, here we go. All right, that should be up now. Yeah,
All right.
that's it. Okay, did everybody see that?
Yep.
Okay, so all right, so what we did this on a large commercial hemp farm near Albany, Oregon. And the idea was the person that was doing this tried 12 different varieties of CBD hemp. And so that made it kind of difficult for us to do any research. So we just took basically two varieties. They're the most popular varieties, and then we went with them. Oh, that's not right. Oh boy, I'm having trouble here. So what we did was we tried 3 different approaches to this. We used what we call full season applications of humic or fulvic acid, either fulvic or humic. And then we had long-term and then where we continued the operation to about halfway through the season, and versus all season. And what was the difference there? The difference was very different situation with the results at the end of the year. Okay. Yeah, this I uploaded a new system. It's okay. So now you can see the pictures. Okay, so that's the field in the distance, and that's me with the blue bioag shirt on. This is hemp. This is hemp grown for CBD. Okay, so and you can see the size of the field. You're talking about 40-acre field, and here we are at harvest time, and all right, next slide. Abstract. So what got me on this was that there was research done in Israel on it was sort of inside, growth chamber research using humic acid. It was kind of simplistic. I mean, they did it was pretty complex work, but how they did it was fairly simplistic. So they used one humic acid product. That's it, of really unknown composition, unknown content. And based on that research, they published a paper, and it did show modification of cannabinoids based on the humic substances. And that's the idea. Okay, so here's a shot. It's right next to I-5, north of Albany, Oregon. And there's the field. And there's a lot of money went into this. Unfortunately, it didn't come back in because they couldn't really move all that product. I mean, everybody had the idea that, they're going to get rich with CBD, but when it came down to it, the market just wasn't there unless you had really prepared, ahead of time and got a contract. So biostimulants and their effect on CBD and THC. So we know the cannabinoid content and cannabis sativa are known to be plant responses to drought and stress environmental factors. The drought stress plants had increased concentration of major cannabinoids, THC, THCA, and CBDA. So that's from some of the scientific literature as far as drought stress, the effect of humic substances on drought stress. We know that's true on most plants, including wheat and everything else, because it increases the sugar content of the leaf, and that acts either like an antidesiccant or an antifreeze. Okay, so this paper that the Israelis did demonstrated that a decreased cannabinoid content from applied claim 12% humic acid product of unknown sources. This actually showed a decreased cannabinoid level from use of humic acid. So my interest was to expand further on this study and apply humic and fulvic acid in a commercial setting, which includes commercial cultivars of cannabis sativa and 30 acres to commercial operation. Outcome was to observe CBD and THC content of the plant when specific amount of fulvic and humic acid are applied. Okay, so that was the whole idea of this whole thing, was to take this a few steps farther and see what really happened. So we used all these different varieties. I won't go into this. It was quite a complex, difficult project. I can see why they didn't want to do this. So we had a control, which is the pink flags, and we had a humic short, what we call humic short. That's 1 gram a gallon of full humics was dissolved in distilled water and foliarly applied from the first week of planting and every 7 days until flowering starts. So that's every 7 days. It's 1 gram per gallon till flowering starts. Total 9 applications were applied. This is foliar application. Fulvic short was full power, our fulvic acid product, 30 milliliters per gallon, were applied from the first week of planting every 7 days until flowering start. 9 applications. Humic long was the same rate, 1 gram per gallon, applied from the first week of planting every 7 days until harvest, all the way through. 12 applications. Fulvic long, same thing, was applied all the way through. The application protocol started a week after the plants were planted in the field, the first week of July. So these things were planted late, and they were already grown in the greenhouse from seed, and they were very uniform, which is part of the deal. So we just let them grow. We sprayed them. And we did the data collection. This paper was done for the International Humic Acid Society. That I presented this last summer. So it's kind of an academic exercise in a way. But so I'm just going to get down to the bottom line on this. So the bottom line, results of discussion. Results vary by hemp cultivar. That was kind of an interesting finding. The cultivar Suver Haze being the most responsive. There were 12 cultivars involved. Plant height and width are sensitive to the type of fulvic and humic applied and the duration of the fulvic humic applied. So it's not just going out there and spraying something. It's like how long, how much, concentrations, that sort of thing. So the 2 varieties we tried was Lifter and Suver Haze. So the Lifter, the fulvic long and humic long will provide the best biomass. That's biomass. Suver Haze, fulvic short and humic long will provide the best biomass. So it kind of depends on the cultivar. In this case, I mean, there's a company here nearby that produces those 12 cultivars. So it's okay, results of discussion. So we go to the results of discussion. Results vary by cultivar. Plant CBD and THC content are sensitive to the type of fulvic and humic acid applied and the duration of the fulvic humic applied. Okay, on the Lifter variety, to increase CBD and reduce THC, used fulvic all the way to harvest and use of humic all the way to pre-flower, okay? And Suver Haze is to increase CBD and reduce THC, use fulvic all the way to harvest and use humic all the way to pre-flower. And that's how we got the highest CBD levels and the lowest THC levels. So the control, you can see humic, fulvic long had, okay, the control had 0.33 THC. Okay, that guy'd be over the limit, see. The fulvic long was 0.26. So he's good. See, and so humic short. And then, so what ends up happening here is humic substances reduce plant stress. So the THC formation is based on stress. It's a stress response. So if you reduce stress at that point, at the flowering, the maturation of the bud, it's not going to have as high a THC level. Simple as that. So if you have hemp and you want to keep your THC level down below the number or the magic number, then you'd want to reduce stress through the whole growing season, and that would be the use of humic substance, humic acid, fulvic acid. Okay, so so this is an important tool for people that are growing hemp, because a lot of my friends in Hawaii, they grew hemp in Hawaii. Hawaii is a great place to grow hemp, but it had high THC and they had to burn their crops. So, so we had to find a way, and other people have had that problem all over the country. So how can we modify that? Okay, we can do it by reducing stress, and that's the use of fulvic and fulvic acid. Okay, so so that's what we did, and of course here's all the data behind it. THC percentages, CBD percentages, fulvic long, that was giving us the highest CBD content, about a 23% increase, and fulvic short, 10% increase. So you have a bunch of different numbers here, and you can see it in these charts. The control had the highest THC, and the fulvic short was lower, fulvic long, lower still in THC, because we were reducing stress. Now, CBD levels, fulvic long, well, it also increases the CBD levels. See, so it's sort of a juggling act between CBD and THC. But anyway, we know that this is the protocol to follow to increase your CBD and reduce your THC levels. Okay, now on the opposite side, you'd want to, you'd really want to, if you're going for high THC, you can see the high THC levels are more associated with reduce and the plant weight too. So if you have THC levels and weight, plant weight, so that's your yield. And here's your treatments to control, and fulvic short, fulvic long, humic. And so you'll see some major differences here. We have a much higher CBD level, is the mean here. Anyway, I don't want to get into the super details on this because we don't have time. So the discussion of the results. So bottom line, I've already pretty much mentioned that. Many cannabis growers over the years, I mean, back 50 years ago when well, it's 53 years now, I guess, when I grew my first crop. But we believe that the plant's stress factors increase the potency of the cannabis. This was a belief. This is like something we kind of believe, that it was a stress. A practice called flushing was widely employed in both media or hydroponic production to leach out the salts and reducing nutrient availability, causing nutrient stress. Also, many would reduce or eliminate irrigation at the budget. So you're reducing the water, which is a good idea. You're leaching salts, or if it's outside production, you're not fertilizing. You don't fertilize at the end. This also increased the overall quality factors. The work in Israel verified this effect and carefully controlled environmental growing and sensitive testing. Our work took the concept to actual large-scale field production of hemp bred and grown for CBD production at low THC levels. So I went over that a little bit. The THC levels have to be below 0.03% and be harvested. They had to be tested in the field by the state, and then they say, okay, you're good, or you're not good, you got to burn it. So that's the idea. It's completely arbitrary too. I mean, there's no reason for this to begin with. But it's an interesting exercise. So that's about it. Here's the references. So, I'm just going to try to sum this up. You need to keep nutrients down. Like if you want high THC, you want to reduce your use of any nutrient substance. You want the plant to stress naturally. You want it to kind of, run out of moisture a little bit, be stressed. run out of nutrients. Yellow, the bottom leaves, the large leaves turn yellow and drop. Okay. Now, if you use fulvic acid at this point, that reduces stress. And then those things don't happen. You may end up with a nice green plant that's not doing that. Now, a lot of people are really worried. They start seeing the leaves turn yellow at the end of the growing season, and they get worried and they call me, should I fertilize now? Should I fertilize? No, absolutely not. I want all those leaves to turn yellow and drop off. That's not what I'm not interested in those leaves. Okay, I'm interested in the bud. And so, okay, it's all about stress. Now, why is that? And by the way, we used a new test method to test for THC and CBD, the GEMA equipment, which was a quick test for CBD and THC. But what's causing this is the stress hormone, abscisic acid. So abscisic acid is called abscission. That's how the leaves drop off. They turn yellow, they just drop off. That's it. Abscission. Now you can buy it. I'm not suggesting this, but they use there's synthetic versions of abscisic acid they use to drop, for instance, olives off of trees, like ornamental olives and things like that. It causes abscission. Well, that happens naturally, if you don't try to prevent that. So if you want to grow CBD and you want to have low THC, prevent stress. If you want high THC, don't prevent stress. Let the natural cycle take its place. Let those leaves turn yellow. I know it's a fine line, all right, but that's what the research shows, and we will continue to do this research. I guess that's the end of the slide. So we're going to continue to do this research and get it. It's a matter of fine tuning, really. Now we've got some other research we did that has to do with THC. Okay, so what's the limiting factors of THC production? Because here in Oregon we have both. We have large scale production of hemp for CBD and cannabis for THC. And so we've been working with some new products based on that. Can you see me now?
Yep.
Okay. So is there any questions in the meantime?
Let me check here in chat. Do you want to also just touch on your background with humic and fulvic acids? You studied quite a bit more than most other people I'm aware of, and maybe not everyone is aware of your background on that.
Okay, well, yeah, I started out as an entomologist. I went to University of Delaware, University of New York, blah, blah, blah. So I started out, my interest was biological control and IPM. And I was like young, idealistic. There's got to be a way around pesticides, and there is. And it's biocontrol, biofungicides, bioinsecticides. And so my research over the years started with working with seed potato producers in Montana. And I heard about reading in a magazine, somebody had something called clodbuster humate. So what's this? I had a hard concrete-like soil on this land that I owned in Montana. So I got a little sample, I tried it. Hey, this stuff works. And then we tried it on large scale fields, like seed potatoes, 40-acre fields, split a field, 20 acres with, 20 acres out, without. And what we saw was no or very little insect damage right to the row where we put this humic acid or humate. It was a New Mexico humate. So, and then the next row over, the unapplied area, it looked like Swiss cheese. And wow, I'm a young idealistic guy. This is back in '73. And whoa, I think we got something here. What's this? We just put this, looked like dirt. We put it on the ground, 400 or 500 pounds an acre. And what? This could save the world. So that's how we felt, or how I felt at the time. Of course, academia extension agents are saying, oh, it's snake oil. Yeah, that doesn't do anything. You know? Well, I knew it did. I mean, once you see something with your eyes, I don't care, you know, I mean, maybe some people will listen to the, you know, BS, you know, but I didn't. I knew there was something to it. So that's where I started. So I went from entomology to more soil management agronomy as a means of reducing pest pressures. And we didn't know, but I set my sights on that I was going to figure that one out in my lifetime. Like, what's happening? And so that's what I've been doing for 50 years. And I'm a member of the International Humic Substance Society, the Humic Product Trade Association. I follow all the research. Now, when you talk about humic acids, I mean, my original interest before I got into trying like a humate was biodynamics. And I farmed in Pennsylvania, and I was close to the Biodynamic Association, and I had Amish and Mennonite neighbors that were using a biodynamic compost, and I tried it. I was still in college working on an advanced degree, University of Delaware. So I was doing research on the family farm, and I was seeing incredible things, you know, that they don't teach you in the university, but you can't dismiss it. There it is, right in front of you. And so every year since, I've been doing research, and what I found out, okay, what's the secret of humic substances and insect damage? Now, a lot of people that use our products, the BioAg products, the Full Power, etc., report we don't have any insect problems. Now, we have big gardens here on my farm here in Oregon. We have 20 acres, and we do all kinds of test plots and vegetables, and you name it. And same thing. I mean, we grow cannabis, you know, we grow hemp. You know, I have a medical grow here. I don't see any insect problems, and our customers say the same thing. I mean, you might get some mite problems, you know, at certain times in season inside, but not outside. We do it outside and inside. We do hydroponics, we do soil, we do it in all kinds of ways. So that's where I'm coming from. You know, I come from the pest control, pest management, biocontrol, entomology. And there's a connection. There's an incredible connection. So what did we find out? We found out, because the science didn't exist at the time, that could explain that. So it didn't exist. Okay. So now we have DNA. We have transcription profiling. We have pathways, genetic pathways. Okay. So we know that humic substances, certain components of humic substances, mainly phenols, and you can get down to tannins within the phenols, activate pathways that turn on insect and plant pathogen resistance. So when exposed to these components, the plant reacts by turning on its defenses. So we can pre-defend the plant that way. So we give it what it needs to take care of itself. Obviously, you know, people have been farming and growing cannabis for 10,000 years, longer than that, who knows, you know, but it's a long, long time. And there wasn't any pesticides or synthetics or anything like that. So what were they doing? You know, how did that work? So we know it worked. The plants don't need any help. They wouldn't evolve. They wouldn't be here today if they needed help, because there's natural mechanisms. And when we provide humic substances, and that can be compost, that can be humates, that can be earthworm castings, there can be a lot of things. But if they have the specific functional groups, see, you're talking about a carbon source, but it's really species of carbon. So what kind of carbon are we talking about? You know, back in the day, like the researchers, university guys would say, oh, that humate can't do anything. It's only a carbon source, and you're not putting enough carbon on the soil to do any good. How can that work? No. It's specific carbons, like polyphenols, carboxylates, carbonyls, and you can tell the quality of humic substances based on the functional group distribution, and by experimentation. So this just, and the work that I got was from going to international meetings and meeting with some German researchers from Humboldt University of Berlin, who took our product and traced the genetic pathways that were activated and determined which they are. I mean, named, numbered pathways. So we know what pathways are activated by what components in humic substances to turn on the plant's defenses. It's not just defense against insects and pathogens. It's also activation of what they call plant growth regulator pathways. In other words, instead of buying a product that you spray on a plant that has plant growth regulators, let's say a kelp product or a synthetic product or a whole bunch of other stuff, we're turning on that function in the plant. The plant's going to make its own based on that genetic pathways. And I can tell you what the pathways are. We know what the pathways are. Of course, that's kind of meaningless at a certain level. But yeah, so that was 50 years in the making, that we went from it's snake oil, it's carbon, to genetics and activating pathways and functional groups and all that stuff, because the science didn't exist. And all that revelation came from younger researchers, PhD researchers in Germany. And there's more. I can tell you there's more. These same components stimulate fungal reproduction in the soil. And other published papers I've done was using humic substances, either fulvic, humic, or both. And what happens to soil microbial distribution? So it rapidly, within 60 days, goes from, if it's an old worn out soil like here in parts of Willamette Valley, it goes from bacterial dominance to massive fungal dominance. So it's like, I don't know, it's like priming the pump, okay? Now, these soil microbes and fungi and mycorrhizal fungi, they produce humic substances. They produce the humic substance, like BAM, mycorrhizal fungi, like our product BAM. Actually, this is fairly recent research, produces humic substances in the root zone. Okay, but sometimes they need some help. So that's we see sort of a priming effect, and then things go different. The soil becomes structurally improved, crumb structure versus like no structure, basically. And so then you have better, aeration, better, easier root growth, all kinds of good things happen. Do you need a product like, a commercial humic product like what we produce? No. It's either our stuff or a lot of work. I mean, you can make compost. We're going back to biodynamics, or even ancient Hawaiian agriculture, or even ancient Mayan agriculture. They had their methods of producing humic substances. Okay, but see, nowadays, the way we farm now, industrial style or hydroponics or other things, it makes the use of commercial humic acids like our full power Fulvic, for instance, crucial, because it's the living bridge between the dead root and the dead mineral matter of the soil and the living plant root. So it's that interface, and it's crucial for microbial activity. I'll just say it's crucial for cellular function. It doesn't matter what cell, human, plant, fungi, whatever. It's about cellular function. It's about ATP transfer. It's about basic cellular functions, mitochondrial activity. It doesn't matter whether it's blue-green algae, because I work with big blue-green algae growers in Hawaii and using humic substances to increase blue-green algae. That's one of the most primitive things out there. So all the way up to humans, and we have our Wujin-san, which is it's Chinese for black gold medicine. It's been used for centuries in China. And that's for, veterinary remedies, for human supplementation. So all cells need it. It's what's protective against diseases and everything else, and increases vigor and longevity and all that kind of thing. It makes your hair grow. I got lots of hair, right? Lots, for a guy that's 74 years old. So we're talking about cellular activity, and that's what you got to think about. It's not this plant, that plant, blah, 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 or this animal. People say, well, what will it work on? Whatever's got cells, it'll work on. Any other questions?
We had a question from chat. Can you define fulvic acids and the different fractions? You've broken that down really well before, and I think a lot of people haven't heard it.
Yeah, well, okay. So fulvic acid is defined as the acid-soluble fraction of humic substances. So there's no chemical compound called fulvic acid, period. It's a fraction. So when you take a compost or humates and you acidify it, the black stuff or the brown stuff is going to drop out, and you're going to have yellow stuff. In German, it's Geldstufe or gold stuff, right? Fulvic means golden and brass colored in Latin, fulvicus. So it's defined by color. So the fraction is a golden color. So if somebody tries to sell you fulvic acid, it's not golden color. Well, come on, that's like trying to sell you an orange that's red. Is that an orange? It looks like an apple. I don't know. So yeah, it's a color. And then within fulvic acid fraction, there is, well, okay, let me back up. Within humic acids, it's not humic acid, it's humic acids with an S. Fulvic acid is one of the humic acids with an S, along with ulvic, humic, fulvic, and then insoluble stuff called humin, Humin. Okay. So within, what are we looking at within, so the part that plants take up directly, either through the roots or the leaves, is fulvic acid. And it's got a pH of 3 or so. Humic acid, on the other hand, the color goes from, let's say, brownish, gray, sort of light brown to sort of dark brown. Okay, so black is not humic acid. And a lot of people think, oh, this is nice and black. It's not. No. Humic acid is not black. It's not black. That's the waxes and other stuff that's in these deposits. Because humic acid is going to be brown and degrades into gray. Okay, so within fulvic acid, what makes it work? Because not all fulvic acid is the same, I can guarantee you that. It's really the percentage of polyphenols, quinones, carbonyl groups. Like, for instance, in Russia, they figured this out, and they do a functional group analysis, and they'll rate a humic acid or humic substance based on biological activity index. Now, that's where you take the positive functional groups that do good things to plants, that plants benefit from, and you subtract the negative functional groups. Because within coal or some humates, there's what they call methylene groups. That's associated with sulfur, and like hydrocarbons, oil, coal, that kind of thing. That's a negative functional group. So when that tests high for methylene groups, that has to be subtracted from the other number, and you come up with a biological activity index. Like our product is like 86, 88% biologically active. Well, the full power is much higher than that, much higher. But I'm just plain old raw humate with the full spectrum of humic substance. So we've identified what are the fractions that do all the good things, and then you get within polyphenols, which do a lot of the good things, more specifically tannins, which is found in bark, oak bark, and other barks. There's a lot of tannins. It's not just one thing called tannin. Okay, so there's a whole range of tannins And one of those tannins is what activates the M25 or M25 pathway and activates the plant's defenses and things like that. In humans, it affects the insulin pathways and some other chemical pathways. So we have the only lab in the West Coast, believe it or not. Even the state of Oregon has to send out samples, but we have the only lab that can actually test humic substances, and based on the new criteria, which was developed by the Humic Product Trade Association and the International Humic Substance Society, and accepted by AFCO, which is a regulatory body of state fertilizer officials. And so here in Oregon, it's like one of the first states to actually allow us to call it fulvic acid. When we first came here, they wouldn't let us call it fulvic acid, because they say, well, we don't have. But anyway, a lot of lobbying was involved, and they accepted the AFCO and the IHSS technique for analysis, and so we have a guaranteed analysis for fulvic acid. So people say, well, what's the difference between full power, my fulvic acid product, and all these other so-called fulvic acids? Well, look at the color for one thing. Are any of them gold? Maybe. Okay, do they have a guaranteed analysis? No. So how can you make a comparison? You don't even know even the basics. Okay, so ours is a guaranteed analysis, and we've never been called on it either. It's there, just like our mycorrhizal product. Out of 45 microbial products registered in Oregon, only 2 passed. The others were all red tagged, taken off the market. Only 2 made it. It was our VAM product and another company in California. So, when people say, can you compare it to this or that? Yeah, if they have a label. I mean, like for us, our VAM, we guarantee, the spores on the label, and the states checked it, and we have double our claim. So you can't really make a comparison if you don't even know what the numbers are, first of all. We never had, like when we first started, they didn't accept fulvic acid. California still does. So in California, we have to call it humic acid 0.05%, which is their minimum detectable level. They let us do that because they will not. They have not accepted AFCO. It's inconvenient for their lab, But does that stop us from selling it? No. We sell a lot of it because it works. I mean, people aren't buying it based on the label. I don't think people do that more than once. It's got to work. It's got to work. So that it's really breakthrough stuff. It can change the world, because my study of ancient civilizations and agricultural systems, it's very clear to me at this point that these civilizations fail based on the soil property, soil fertility, and their ability to feed growing populations, or with diminished, soil health, soil, humic substances. And usually they're under pressure to farm more ground, and it goes down the tubes. And we're no different. I guarantee you that. I mean, we may have high-tech agriculture here, but guess what? That doesn't matter, because once the soil runs down, humic substances only last 60 days. They start to oxidize. So it has to be a complete cycle. You have to keep building all the time. Building, it's like, it's like Shiva. It's creating and destroying. And that's what's happening in the soil. You have to keep replenishing organic matter, humic substances. But then once you get the soil life going, like the mycorrhizal and other things, they will produce humic substances. Humic substances are produced by fungal breakdown of organic matter. And you get down to the basic definition of humic substances. It's the end product of decomposition by specific microorganisms. And that's mainly fungi, okay? And So that's the key. That's the difference between the Earth and the moon. Okay. So is there any other questions?
You've also done a lot of work with kelp. Do you want to talk a little bit about your work with kelp and kelp testing?
Yeah, I learned about kelp back in the day when I was farming in Pennsylvania. I was still going to college. My Amish neighbors were using kelp. They said, "Hey, Bob, you want to grow good corn? All you got to do is spray some of this kelp extract on your seed at the planting." And I'm saying, I went to college. I went to ag school, university. We never heard of that. But can you ignore an Amish farmer who produces some of the best crops in the area? I mean, you can't blow that off, right? And so I did that. I've been doing it every year since. I just sprayed kelp right on the extract, right on the seed and plant it. That's it. And they were telling me, "That's all you got to do, Bob. You don't need to do anything else." Well, okay. And so that got me into kelp. And now we make a product called Cyto Plus, which is a combination of kelp, soluble kelp extract, humic and fulvic, and in trace elements. So I'm looking at what are the limiting factors. This is the package, right? So it's got the kelp effect, and that's a little bit different. Now, kelp also contains humic substances. So it also contains humic substances, but more. It's got your IAA, your what they call cytokinins, that kind of stuff. So these things go together. Matter of fact, there's a synergistic effect between humic acid products like ours and kelp. So you put them together, they work better than they do individually. That's a synergistic effect. And they also have the anti-drought, anti-frost effect. And there's a high availability of certain trace elements that you're not going to get in anything else. And then again, there's a lot of kelps on the market. So it's not just say kelp. It's got to be the right one. It's got to be processed the right way. Now, for years, I've been using Norwegian kelp. People say, "Well, why don't you get the Chinese, or why don't you get the..." No, I mean, I'm sticking to what I know, works, and it's good, And so that's what we use in our product called Cyto Plus. And Cyto comes from cytokinin, and that's the substance. And all that means is something that stimulates cell growth. They didn't know why. We kind of know why now. So that's the deal on kelp. And we recommend it as a mid-season. We recommend kelp applications at the onset of bud or flowering. It doesn't matter what kind of plant it is. It can be tomatoes, it can be tree crops, but you use that to when the energy shifts from the male energy to the female energy, and then it's applied. And that's like old Dr. Sen used to say. He's the guy that did the research and some of the first work on kelp and what's in it, cytokine. But he used to say to farmers, it makes the plant sexy. It shifts the energy to the female energy. And of course, in hemp and cannabis, and most crops we want, unless it's hay, we want the female part. We don't care about the male part. Like I tell people that, we're not growing hay. That's not what you're after. So we shift that energy mid-season, right at the beginning of initiation of the budding, after the vegetative growth is complete, and then we shift it to by using the kelp and humic foliar spray or soil applied or hydroponic, applied to the nutrient media to get that effect, to pump those buds, let's say. Or make the fruit nice and big. My apples, you see my apples. That's the only thing we use. We just spray it with Cyto Plus. That's it. And I don't have any insect problems. I don't have to spray my apples or my fruit crops or anything, with any kind of pesticide. And we know from other research that kelp gives plants resistance to mites. They don't like it. Something happens. So this goes back a long way. There's an effect of insect repellency or something, from kelp, and it probably goes back to the same, pathways and resistance factors and things like that. So it's the same with people. You take kelp, you take humic substances, you're going to have a high level of resistance. Why? It's a cellular response. Guess what gives you resistance to diseases and pathogens? Things like T helper cells, white blood cells. It's all about cells. I want to stimulate my immune system. And those cells that protect us against these invaders, viruses, viroids, bacteria, whatever, and kelp fits into that. I mean, kelp, look at the Japanese. I mean, they have some of the longest longevity in the world, as long as they stuck to their traditional diet, I'm going to say. In Hawaii, the most healthy people walking around in Kona were Japanese. And basically, they're eating kelp in every meal because it's part of the ingredient in a lot of Japanese cooking. What they wrap the sushi with, that's only part of it, right? So you can't deny that, that there's health benefits to kelp, and also with livestock, feeding it to livestock, along with humic substances. I mean, the two go hand in hand. It's simple as that. I think it's the basis of all life on Earth. It's the difference between, dead sort of unproductive world and a productive world. So that's the story on kelp. But I wouldn't be using it. I mean, it's a timing thing with kelp. I'm talking about plant growth. Now, if you're feeding it to people or animals or fish, that would be a continual feed setup. And of course, I have quite a bit of background, in the aquaponics or the aquaculture thing too. Matter of fact, part of my PhD thesis was coming up with a method of producing aquaculture feed, a high protein ration for, say, tilapia grown in geothermal aquaculture. And so that's where we take lignocellulosic waste. That can be straw, that can be hemp stalks, a lot of different things. And they are fermented with a specific fungi, what they call white rot fungi. And so it can convert like a low protein, something like wheat straw with 2 or 3% protein to like 14% protein. And then that can be fed directly to fish, particularly tilapia love it. So that's because what I was saying is the most expensive part of raising fish is the feed. Well, if we can produce the feed from lignocellulosic waste, which is just huge, huge amounts, then it's going to be a lot more economical, method. But I could never get, investors or people interested in that. I think maybe someday if they get hungry enough, maybe we'll be looking at that. But that's a possibility too. Now we use humic substances in fish ponds, commercial aquaculture, like in South America, we sell our product full humics. They throw it into the pond, shrimp, fish, whatever. And now there's all kinds of research from the same university, Humboldt University of Berlin, about how humic acid does good things for fish. And it's basically the same basic story, except fish live in that environment. They're surrounded. And in nature, fish live in an environment that contains fulvic acid, humic acid. If they don't, they're in trouble. That's when you start getting diseases, and viruses, and all kinds of problems. So this whole thing is just part of the cycle of carbon on Earth, that cycles through cells and organisms. And once you break that cycle, well, that's the end. That's kind of the end point. And just like I tell farmers, if you farm without organic matter additions or humic substances, you're just waiting. You're going to have to go to town and get a job at some point. And of course, over 50 years, I've seen a lot of them go to town and get a job, or drive trucks or whatever. So yeah, And then from a one-on-one deal, everybody here ought to be taking, fulvic acid and kelp. I'm not trying to sell anything. I'm just saying, it's a proven. Well, again, Humboldt University Berlin. Why were they testing my product? Full power. They were screening for longevity substances, things that increase longevity, and they use a indicator organism to do that. And they proved, in the lab, not with humans, because how can you prove longevity in humans? That'd be hard. That a fulvic acid is a longevity substance, increases the longevity, the lifespan of something, the organisms. They use a heterohabilis. It's a little nematode commonly used for that kind of work. So that's what's so crucial. I mean, you can talk about products all you want, but it comes down to humic substances, because the people say, well, the soil isn't good anymore. Like, don't we have to take minerals? Should we take minerals? They tell me there's no minerals in the soil. Well, if there's no minerals in the soil, nothing's going to grow. I mean, there is minerals in the soil, but it has to have something to release them, and that takes soil life. It's not, the soil isn't just something that holds the plant root and you pump salt fertilizer on it. There's more to it. But our program, in the US and the world, is rescue chemistry. So, you use chemical salts, and then you rescue the crop repeatedly with pesticides, fungicides, etc. So that's the program. It makes good money for big companies and for the elite. So yeah, it's a money maker. But they know this, and that's how it works. It's crop rescue. And it's people rescue too. I mean, just watch the news. I'm sure everybody does. I mean, every other commercial is some drug. So that's it. Deprive people of the natural cycles, of humic substances, and then you have to rescue them and their crops and their pets. So I think that sums it up.
Yeah. How can people find out more about the work that you've done and the research that you've done? We can keep going a little bit longer while we're waiting on Murray to reconnect, but why don't you tell everybody, just in case he does connect here in the next moment or two, how to find out more information about your work?
Well, we have a website. Now we have a new guy working on that. We have people that are, doing, we have an incredible amount of research. I wish I could go through my next thing, which was pictures of our new product, which was BioAg Supercell. See,
if you want to,
yeah,
while we're waiting on them, you're more than welcome to.
Yeah, you want to go to bioag.com. It's just BIOAG.com, bioag.com. And no, we're not part of Monsanto. We're the mortal enemy of Monsanto. We survived, they didn't. Okay, so yeah, some of our products. Yeah, okay, he's showing the website. And then we have experts that work for us, and they can answer questions. We have people that are real pros at all kinds of cannabis production, vegetable production, everything. So we have technical reps that can answer a lot of questions, and our website has a lot of questions answered. So yes, and then he's showing the website. And then we have experts that work for us, and they can answer questions. We have people that are real pros at all kinds of cannabis production, vegetable production, everything. So we have technical reps that can answer a lot of questions, and our website has a lot of questions answered. So yes, and then he's showing the website.
Looks like I got a little feedback there. I apologize.
A little feedback. Okay, so are we okay now? I see there's a question there about our micronized fertilizers and silica. When will that be for sale? Okay, well, that's our newest product. we call BioAg Supersil. And it solves the problem of silica. The two major limiting factors on any given field is humic substances and monosilicic acid. Okay, it's hard to work with silica because it's reactive and it ties up and there's problems. So we worked for years on how to get silica into the plant without it combining with other things. While we're working with a company out of Germany, and we're using naturally a microbial produced nano silica. Okay, so that is combined with humic substances. So it doesn't have to be in a soluble form, the silica, if it's in a nano size. And this isn't just nano height; it's the real thing. It's determined with electron microscopy. So plants can take up these nanoparticles and then they digest them, you might say. Put it in a simple way, they can metabolize that.
And what was the name of the product again?
BioAg Supersil.
Awesome. Well, definitely have to give that a try. It looks like Murray finally was able to connect. Well, thanks again, Dr. Faust, so much for joining us. And why don't you tell everybody one last time how to find out information about your teachings?
Yeah, well, if you go on bioag.com, there's a lot of stuff there. We're adding more stuff all the time, as we've done. Yeah, you got 3 or 4 researchers we working all the time, and so we're putting up new stuff because we're tired of this BS that's out there in the marketplace. You know, we just want to get down to the nitty gritty, and we use the scientific method, but we also were open-minded to the interpretation. But it's still the scientific method. It may not be the status quo yet, because regulatory capture and the effect of the big ag, big pharma, and all that stuff. So bioag.com can answer a lot of your questions, and you can call our technical reps, like our technical guy for hydroponics and cannabis is Josh Hedberg. And he'll take calls. I won't necessarily take calls, but I have people that will. And so there's more. There's way more. But I mean, that's just the beginning of what I said today. We have these rhizosphere positive cultures we're working on that work with the VAM. They work together synergistically. It's really, really incredible breakthrough stuff. Signaling using volatile organic compounds, signal all kinds of things in the plant. It's really like the next level, really. Awesome. It's real organic farming. Yeah, it's the real.
We'll definitely have to get you on the podcast soon to learn more about some of the cool new stuff you've been doing.
Yeah, really, because it's crucial stuff.
Awesome.
And I can show you the pictures.
Thank you so much. We'd love to have you back on the podcast and give us a longer explanation of that part. And I really appreciate you taking the time to join us for the conference.
Thanks, Steve.
Thank you.
We'll do it again. Yeah.